Friends, welcome to Biology Made Easy. Today in this video, we'll discuss about ribosomes. Ribosomes are the smallest organelles of the cell, and they were observed only by means of electron microscope because they are so small in size. So we'll discuss about the ribosomes. The ribosomes were first discovered by Robinson and Brown in case of plant cells, while in animal cells it was observed by George Palade in case of animal cells, and also he coined the term ribosome. That is why ribosomes are also known as Palade particles. Ribosomes are known as Palade particles. Ribosomes are such organelles. They are present in both prokaryotes and eukaryotes. And in case of prokaryotes, their number usually varies from 20,000 to 30,000 per cell. While in case of eukaryotes, their number is almost 500,000. Almost it is 500,000. And in case of eukaryotes, the ribosomes may be present either in a free manner or they may be attached to rough endoplasmic reticulum. That we see this may be free in the cytoplasm or they may be attached to endoplasmic reticulum, attached to rough endoplasmic reticulum. So if they are attached to endoplasmic reticulum, they are attached by means they are a bigger unit by means of ribophorins 1 and 2. Bigger unit by ribophorins 1 and 2. So whether they are free or attached, their structure is same. Now in addition to, in case of eukaryotes, in addition to that free and attached to rough endoplasmic reticulum, in mitochondria and chloroplast also the ribosomes are present. That is, in eukaryotes also you will find it in case of mitochondria and the plastids. And the ribosomes which are present in this, they are usually known as organelles ribosomes to distinguish them from the cytoplasmic ribosomes. Now, what about the structure of the ribosomes? We will discuss about the structure of ribosomes. Ribosomes are usually membraneless. That means there is no membrane surrounding it. So it is membraneless. It is the smallest organelle. Then it is a ribonucleoprotein. It is a ribonucleoprotein. That means it consists of RNA and protein. That is why it is known as ribonucleoprotein. And because more amount of RNA is there, that is also acidic in nature, ribosomes. So it is mostly made up of ribonucleoprotein. And particularly in case of eukaryotes, the ribosomes are synthesized. They are synthesized in the nucleolus. That means the nucleolus is the factory for synthesis of ribosomes. And particularly in the ANOR part, nuclear organizing region of SAT-chromosome. In the SAT-chromosome, there is one nuclear organizing region which we have discussed in the study of chromosomes. So there is one rDNA, and this rDNA it transcribes rRNA. rRNA is ribosomal RNA that takes part in the synthesis of ribosomes. So this is mostly in the eukaryotic cells. But in the prokaryotic cells, they are synthesized directly in the cytoplasm because there is no nucleus or nucleolus. Now, this is one aspect. Now, the second aspect is each ribosome has two subunits. One is a bigger subunit, and one is a smaller subunit. And usually these two subunits are always free from each other. They will combine together only at the time of their function, that is protein synthesis, only when the magnesium concentration is less than 0.0001 molar solution. When the magnesium concentration is less than this, then they will combine together. If it will be more than this, then they will give rise to dimers. Now, the two subunits which are present, they have different structures. The bigger unit has a stalk, one protuberance, one valley, and one ridge. While the smaller unit has a base, one head, one cleft, and one platform. And both of them are united together. So if you draw the structure of this bigger unit and smaller unit, the bigger unit will look like this. This is the bigger unit, and this bigger unit, in the bigger unit, this is the stalk, then this is the protuberance, then this is valley, and this is the ridge. This is the bigger unit. Now the smaller unit looks like this. This is the smaller unit. It will be slightly bent. This is the smaller unit. And in case of smaller unit, this is the base, this is the head, this is the cleft, and this is the platform. And this is the smaller unit. Now, in case of both, whether it is the eukaryotic or prokaryotic, usually they consist of two units. Now, usually in case of the eukaryotes, the ribosome is known as 80S, and here the S means sedimentation coefficient, or you can call it as a Svedberg unit, which is a measure of size and density. This is very important. That means S means Svedberg unit, which is a measure of size and density. Now this eukaryotic, which is 80S, it has two subunits, and these two subunits, this is one is 60S, the bigger unit, the smaller one is 40S. And the bigger subunit, it consists of 28S plus 40 proteins. While the 40S, it consists of 18S, 5.8S, and 5S, and consists of 23 proteins. That means here how many types of RNAs there? The RNAs are four types. Four types rRNA, plus here 40, here 23. That is plus 63 proteins. And here the RNA protein content, is here 60:40. That means here you will get more RNA and this is protein. But in case of prokaryotes, that is the 70S. Now in case of 70S, the bigger unit is 50S and the smaller unit is 30S. And the bigger unit, it consists of 23S and 34 proteins. While this consists of 16S and 5S, and there are 21 proteins. That means altogether there are 3 types of rRNAs. These are all rRNAs. These are all rRNAs. Ribosomal. All of these are ribosomal. This is 5S. So that means altogether it consists of 3 types of rRNA. 3 types of rRNA, and proteins are 34 and 55. 55 types of protein. And here it is 60 and 40 RNA protein, but here it is 40 is to 60. That means the RNA is, this is the RNA is 40 and 60 is protein. That means both of them are they consist of RNA and protein, but in case of eukaryotes, the RNA contains more in comparison to the prokaryotes. And these are the different types. These are very, very important from examination point of view. So, except this 5S, the remaining 3 are synthesized in this, that is the N or part SAT-chromosome, while 5S is synthesized in the cytoplasm. So, this is about the structure of this. Now, the ribosomes, they may be present either freely in the cytoplasm. So in that case, that is known as monoribosomes. Monoribosomes, and sometimes they may be attached in the form of a chain at the time of protein synthesis. Likewise, so many ribosomes will be attached in the form of a chain or in the form of a necklace, which is known as polyribosome or polysome. This is known as polyribosome or polysome. Now another important thing is, inside this bigger unit, there is the enzyme, because the main function of this ribosome is protein synthesis, and for protein synthesis the enzyme is peptide transferase. So the peptide transferase enzyme is present in the bigger unit. The bigger unit contains the enzyme peptide transferase, and this peptide transferase enzyme is actually not a protein enzyme. This is an RNA enzyme, and this is a 23S rRNA. 23S rRNA in case of the prokaryotes, and in case of bigger the eukaryotes, it is 28S. That means this is enzyme. That means peptide transferase is one rRNA. So here it is enzymatic in function. So that is present. In addition to that, the ribosome has 4 sites. It has 4 sites. Now one side is for during protein synthesis it will be attached to the mRNA. So one is mRNA binding site. Then the second one is A or amino acid site. Amino acid site for incoming amino acid attachment. That means the incoming amino acid will come and will be attached to that A site. Then another is a P site. P site, or it is also known as peptidyl site, or you can call it a donor site, which donates the amino acid so that the chain will be elongated. And the fourth one is E site, or you can call it exit site, through which the used tRNA is removed out of the ribosome. That means the tRNA removed at this site. So this is the structure of the ribosomes. Now if we discuss what are the important functions of ribosomes. The ribosome is the factory of synthesis of proteins. That means it is known as factory of proteins. That is the most important function. So the most important is known as protein factory, or sometimes it is also known as the engine of the cell, or cellular engine, because it synthesizes all the types of proteins and the enzymes. Then the ribosomes may be free or they may be attached to endoplasmic reticulum. So though it synthesizes proteins, the proteins which are synthesized in the free ribosomes and in the attached ribosomes, their destination is different. So if you write that one, that means the attached ribosomes to rough endoplasmic reticulum. Usually they synthesize proteins. Proteins to be transported through the endomembrane system or through the jaw system. That means they will pass through the jaw system or endomembrane system, and they are usually mostly the secreted proteins, also integral proteins, integral membrane proteins, and also lysosomal proteins. And those which are free, let me say free ribosomes. The free ribosomes, the proteins which are synthesized in free ribosomes, they are mostly used in the cytoplasm or cytosol proteins, then peripheral extrinsic proteins, then proteins for mitochondria, chloroplast, then the peroxisome, and also nuclear proteins. That means the protein synthesized in the attached and free ones, their destination is different. Now another very important function is, since ribosomes, so they synthesize proteins required by different organelles and different parts of the cell, who will give that signal to which one it will go? So inside the ribosomes there is a special type of protein known as chaperon. Chaperon is a special type of protein which will fold the protein in a specific manner, fold the protein in a specific way, and give signal to which organelle it will move, and give signal for its transport. So these are the important functions of the ribosome. I hope now a very clear understanding of the ribosomes. Any question that is asked on this aspect of ribosomes, clearly you will be able to answer all the questions. Wish a very bright future. God bless you.